Chapter 8, Cellular Respiration. Here we come across our second biochemical pathway. It is very similar to photosynthesis in that it is a series of redox reactions. However, this process isn't only limited to autotrophs. This process occurs in heterotrophs as well. So even autotrophic organisms and heterotrophic organisms will carry out respiration. So basically, respiration involves taking that sugar that was made during photosynthesis and it breaks it down to release energy. The equation for cellular respiration is the reverse of what we saw for photosynthesis. And that initial step of glucose breakdown is called glycolysis. So cellular respiration is a redox reaction, and it requires oxygen. Oxygen will be our final electron acceptor in this case. So the oxidation of substrates such as glucose is a fundamental part of cell respiration. So here you can see our equation. We have C6H12O6, 6O2 yielding 6CO2 plus 6H2O plus energy. So here we see glucose is going to be oxidized, and carbon dioxide will be oxidized, and oxygen and water are going to be reduced. We do have two coenzymes assisting in the oxidation process. That would be NAD plus and FAD. These are both coenzymes that assist in oxidation during cell respiration, and that they carry high energy electrons to the electron transport chain. These two molecules are very similar in role play. They are different molecules, but very similar in role play to that of NADPH in photosynthesis. But however, do not confuse them, as this is a new biochemical pathway. So cell respiration produces ATP, and we know that because we are looking at cell respiration and we're producing that energy molecule ADP, where you have an ADP plus a phosphate coming together to form ATP, our organelle of interest here is the mitochondria. Both plant cells and animal cells do have mitochondria. So essentially what we have here is we get oxygen from the air, and oxygen and glucose enters the cells, which release water and carbon dioxide gas, and you get the glucose from the food. So glucose comes from the food we eat. Cell respiration has 4 phases. 3 phases occur in the mitochondria. The 4 phases of cellular respiration include glycolysis. Glycolysis, break the word down, glyco and lysis means splitting of sugar. Glyco, sugar, lysis, to split. This is an anaerobic process that occurs outside the mitochondria, so you're in the cytosol of the cell. When oxygen is available, then you will have cellular respiration taking place. So oxygen is available. This would be true aerobic respiration. And the end product of glycolysis will enter into a preparatory reaction called the prep reaction. And that occurs in the matrix of the mitochondria. And that end product of glycolysis, called pyruvate, will then be broken down to a 2-carbon acetyl group. It will be that acetyl-CoA group that is going to enter into the citric acid cycle, which is now taking place in the matrix of the mitochondria. And it's there where you're going to have NADH and FADH2, our 2 electron carriers, being produced. Those electron carriers produced during the citric acid cycle are then going to take those electrons to the electron transport chain, or the electron transport system. And in photosynthesis, we learned that the ETC is a series of biological membranes that transports high-energy electrons and uses that electrons to pump hydrogen ions across the gradient, to which then that is coupled with chemiosmosis, where you use that established hydrogen ion concentration gradient to produce ATP. So the ETC is going to have NADH and FADH2 give up their electrons, so these molecules will become oxidized here to the chain, and energy will be released to produce ATP. So if we look, here are the 4 phases of complete glucose breakdown. Basically, here we have a blood vessel, and from the blood we get out oxygen gas being released from hemoglobin, and we get blood sugar being released out, and that enters the cell. Now, oxygen gas can simply diffuse across the membrane, but here we have that glucose molecule coming in through a channel protein, so that would be facilitated diffusion. Glucose will initially be broken down into pyruvate in the cell's cytoplasm, or the cytosol of the cell. We do get some energy out of there in the form of ATP. We also get out some NADH. Because oxygen is available after glycolysis, then that pyruvate molecule will enter into a preparatory step in the mitochondria, and that is where the citric acid cycle and the electron transport chain will occur. In addition to that, we do get out more ATPs. So we get 2 ATP out from the citric acid cycle, and we get 32 to 34 ATP out total from the electron transport chain. I'm going to go over these reactions as we cover the lecture notes here, but we will definitely go in more detail in class. I'll write them out on the board for you. So as we cover these reactions, keep a space in your notebook so we could write them out into your notebook under my guidance and help. So carbon dioxide and water are produced during glucose breakdown. So in glycolysis, it takes place outside the mitochondria, specifically in the cytosol of the cell, where the glucose is going to be broken down into 2 pyruvate molecules. Pyruvate is also known as pyruvic acid. So initially in glycolysis, our inputs are glucose and 2 NAD+. And we also have 2 ATP being input. The outputs are 2 pyruvate, 2 NADH molecules, and we have a net gain of 2 ATP because we have coming out 4 ATP total. So here is that reaction. Here is glycolysis, where you have glucose coming into the cell. Glucose gets converted to fructose, and then you have 2 ATP coming in to make that molecule fructose diphosphate, meaning it has 2 phosphate groups. So this initial step here, glucose is going to be converted into fructose diphosphate and then into G3P, because that fructose diphosphate will be split into 2 G3P molecules. This step here is called the energy investment step because we are putting in energy. Now those G3P molecules are going to go through the next phase of glycolysis, which is the energy harvesting step. And in the energy harvesting step, you're going to have generation of an NADH molecule per G3P, and we're going to have 2 ATPs coming out per G3P going through. So now everything will happen twice. So initially we had going in glucose and 2 ATP. Coming out, we have 2 pyruvate, we have 4 ATP total, but a net gain of ATP of only 2, because 2 are initially put in, and we have a net gain of 2 NADH molecules. The 2 molecules of pyruvate are the end product of glycolysis. These ATPs are produced via substrate-level ATP synthesis. And basically in substrate-level ATP synthesis, you have an enzyme that is going to take an intermediate molecule and lose a phosphate group from it to form ATP. So there you could see that molecule here with that weak phosphate bond that's represented by the squiggly line loses that phosphate group, and you have a phosphate here, so you get that one molecule, and that free phosphate will be attached to ADP to form ATP. So that's substrate level ATP synthesis. So we have that occurring here, 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 and here. Now, if oxygen is available following glycolysis, so this process occurs whether oxygen is available or not, but if oxygen is available after glycolysis, the fate of pyruvate will be cellular respiration, where those glucose molecules are going to go into the prep reaction. And in the prep reaction, this is necessary before anything enters into the citric acid cycle. So the prep step occurs before the citric acid cycle, and basically you take that 3-carbon pyruvate molecule and convert it into a 2-carbon acetyl group, and oxygen gas is given off. So in the prep step, the 2 pyruvate molecules are going to combine with 2 coenzyme A molecules, and in that process, you convert to 2 acetyl CoA molecules, and you also get off 2 carbon dioxide gas molecules. In that step there, 2 NADP plus come in and are converted to 2 NADH. NADH is an electron carrier molecule that's going to go to the ETS system, and will be oxidized to help produce ATP. So, mitochondrial structure, very important. So, it is a membrane-bound organelle. We have both an outer membrane and an inner membrane, and the inner membrane has these folds called cristae, and the folds are for the increasing the surface area, because it's on the cristae, on these folds of the inner membrane, that is where the electron transport chain will occur. Now, inside the inner membrane, you have a space, kind of like the stroma of a chloroplast, except the space of the mitochondria is called the mitochondrial matrix. The mitochondrial matrix is the location of both the prep reaction and the citric acid cycle. So here you see a cartoon drawing of what the mitochondria look like. Down here we could actually see an electron micrograph. So this is 45,000 times the original size. So in the citric acid cycle, it's the final oxidation of glucose products. The citric acid cycle is a cyclic metabolic pathway, a lot like the Calvin cycle of photosynthesis, and it's going to occur in the mitochondrial matrix. It produces a lot of NADH and all of the FADH2 molecules that are going to carry electrons to the electron transport chain, because it's in the electron transport chain where you're going to have your bulk volume of ATP being produced. So in the citric acid cycle, your inputs are 2 acetyl groups, 6 NAD plus, 2 FAD, and you get 2 NADP plus 2 phosphate groups. The outputs of the citric acid cycle are going to be 4 carbon dioxide molecules, 6 NADH, 2 FADH2, and 2 ATP. And here's what that is occurring during the citric acid cycle. So initially you have acetyl CoA coming in, acetyl coenzyme A coming in, and it's bonding with a 4-carbon oxaloacetate to form the first stable product of the citric acid cycle called citrate. The citrate is then converted to, citrate will then be converted to succinate, and what you get is NADH here, and you get an NADH molecule here being produced. You also have an ATP being produced. When succinate gets converted to fumarate, you get out an FADH2 molecule. So that's another electron carrier molecule. And then fumarate will get converted back to the starting reactant oxaloacetate, and in that conversion there, you get out another NADH molecule. Now, at the end of glycolysis, you had 2 pyruvate molecules. So again, this will occur because each pyruvate ending glycolysis will need to go through this cycle. So each pyruvate will go through the prep step to form acetyl CoA, and each acetyl CoA will come in and go through the cycle again. The citric acid cycle is also known as Krebs cycle, capital Kreb', named after a gentleman, the scientist who discovered it. So the second acetyl CoA will come in, bind with oxaloacetate to form citrate, and then citrate will be converted to succinate, and out of that you get NADH, 2 of them, you get 1 ATP. When succinate gets converted to fumarate, you get out an FADH2, and fumarate will go back to the starting reactant, oxaloacetate, you get out another NADH. We will review these again in class. And then finally, all of those electron carrier molecules, the NADH and the FADH2s, will go through the membranes of the mitochondria, the cristae, the inner membrane foldings, to produce the ATP. So the electron transport chain is located in the cristae of the mitochondria, and it is a series of carriers that pass electrons. Many of the carriers are cytochrome molecules, a protein that has tightly bound heme group with a central atom of iron. NADH and FADH2 are recycled to pick up more electrons to bring back the electron transport chain. And we'll look at that in class later.